Hello students, I'm back with a new tutorial video. In today's video we will discuss points related to plant tissue culture. Let's start from defining what plant tissue culture means. Plant tissue culture is in vitro cultivation of plants or plant parts on artificial media with aseptic condition. From this definition, we should give a special emphasis to three terms. The first one is in vitro, which means laboratory, which indicates the activities are performed in laboratory. C-count artificial media, that means the activity needs preparation of different media which contain necessary nutrients. And the last one is aseptic condition, which related to maintenance of cleanliness, and which is the major challenge in maintaining it. The other important question will be how this technology is possible. That means, how do any plant cell give rise to the whole plant? The answer for this question will be found in unique features of plant cell called totipotency. Before proceeding to the activities, let's first see the history when and by who the technology starts by. In 1898, Gottlieb Haberland, a German botanist, made the first attempt to use the in vitro method when grow plant tissues. The cells he used were varied, palisade tissues coming from the leaves, the piths, the epidermis, and epidermal hairs. This initial experiment was fruitful for several months. However, the cells did not proliferate further. Despite drawbacks, he made several predictions about the requirements for cell division under experimental conditions in 1902. This have been confirmed with the passage of time. Haberland is thus regarded as the father of tissue culture. Over the years, many experiments used similar parameters. In these experiments, the cells survived for a longer time, but they also failed to proliferate. The first root tips were cultured in 1922, and by making use of subculturing, maintained their cultured roots for 20 weeks. In the 1930s, it was recognized that B, vitamins, and auxin, IAA, were key components in growing root cultures using the tissue culture method. Over the following century, many similar experiments were conducted and researchers began identifying the most important parameters that should guide our tissue culture process today. The other important topic to be covered in this lesson will be laboratory design and organization. A lot of you might be curious about how you can set up your own tissue culture laboratory. What are the requirements? What equipment do you need to have? Where you need to be careful while planning a setup? In order to set up your plant tissue culture laboratory, it is important that you carefully choose the location. The sterile environment of a tissue culture lab is prone to contamination through air vents. You should try not to build it close to a facility which deals with microorganisms, bacteria, fungi, and so on. You should also try that it should not be in the close vicinity of other laboratories which handle insects. Another important point is that the lab should be away from storage places which store seeds and old plant material. However, if you cannot avoid these, then you need to take extra precautions to keep the lab free from contamination. The basic rule essential in the planning of an efficient and functional laboratory facility is the order and continuous maintenance of cleanliness, because all operations require aseptic conditions. A general guideline for setting up a facility is to focus on a design that different units and activities need to be arranged to make operational steps possible with the least amount of cross-traffic. For any tissue culture lab, there are certain basic requirements or facilities that you need to install for efficient functioning and experimentation. These are 1. Washing area. 2. Media preparation area. 3. Aseptic transfer area. 4. Culture/grow room. Let us dive a bit deeper into these areas of a plant tissue culture lab. Washing area. You need this area to wash the explants or other plant materials. You also need this to clean all your beakers, cylinders, and flasks which you will use for preparing media or measuring ingredients. This area consists of large sinks and draining boards. This area also has access to distilled water with space for drying. Thus, it facilitates the preliminary cleaning to kill the microorganisms present on the surface. The conventional method for cleaning laboratory glassware involves chromic acid sulfuric acid soak, followed by thorough washing with tap water and subsequent rinsing with distilled water. However, due to the corrosive nature of acids, it is recommended that for routine procedures glassware should be soaked in a 2% detergent cleaner for 16H, followed by washing with 60-70C hot tap water, and finally distilled water. However, there are some precautions you should be taken during acid cleaning. First, handling of acid and processing of glassware in the acid should be done in a fume hood. Second, Never add water to acid, but for dilution, acid should be added slowly to the water while stirring the last one. After acid cleaning, if the solution in the acid becomes dark-colored, it should be discarded. However, with the availability of a wide range of reusable plasticware, it is recommended that these should be washed with mild detergents followed by a rinse with tap water and then distilled water. Media preparation area. You need this area for preparing culture media recipes and for storage of chemicals and glassware. You can use a large area for media preparation with benches. This area also has hot plates, pH meters, weighing balances, and water baths. You can also choose to have a separate area for the media dispensing unit, which is connected to your media preparator or autoclave present in the preparation area. The important thing to know is the equipment we need for media preparation, which are magnetic stirrer, analytical and top-loading balances, pH meter, refrigerator with freezer or a separate freezer for storing stock solutions, plant hormones, and other chemicals, water purification system along with the space to store the purified water, equipment for sterilization of media, glassware, and instruments, pressure cooker or autoclaves for small lab setup, automated media preparators for research, medium and large-scale commercial tissue culture setups. Media preparation is a crucial step, and you need to develop a system to have a step-by-step routine for preparing media and careful measurement of chemicals. Transfer area. In ideal conditions, a separate room should be made next to the media preparation area or next to the area for the media dispensing unit. However, you need this area to have complete sterile conditions in order to transfer explants to media containing culture vessels. So the activities for this room are transfer of explants to growth media, for subculturing, for multiplication transfers, and for transferring multiplied shoots to rooting media. One of the most important key points for this area is that it should always have positive pressure airflow. You need positive pressure airflow to make sure that air can leave the room without circulating back in. Another important thing for you is to construct this area in a way that all the surfaces stay free from dust and microorganisms. You need to always clean this area thoroughly. For making transfers in this area, you need to install laminar flow cabinets as well. This is to ensure constant flow of bacteria-free filtered air, and you do subculturing and other transfers within these cabinets. You need this also to prevent non-filtered air and particulate matter from settling down on the surface while making transfers. So this works by forcing air into the laminar unit through a dust filter and then passing it through a HEPA filter, high-efficiency particulate air filter. This is known as a flow unit, and you have two types. Vertical flow unit. The air from HEPA filter is directed downwards. Horizontal flow unit. The air from HEPA filter is directed outwards. Culture/grow room. After transferring explants and shoots to the media, you need to store them in ideal conditions for efficient growth and development. This means that you need to provide controlled temperature, humidity, air circulation, light quality, and duration of light. You should know that an automated climate and air filtration system works best for regulating these conditions. These environmental factors are important for the plant cells to grow and differentiate into healthy shoots and roots at later stages. These specific conditions differ with each plant species. Hence, you need to be careful with providing the necessary growth environment for optimum development of tissue culture plantlets.